Mesdames, Messieurs, bonsoir ou rebonsoir à ceux qui étaient déjà présents hier soir pour la première conférence. Bienvenue à cette deuxième conférence des colloques Wright pour la science, 17e édition cette année. Ça fait effectivement déjà 30 ans que la fondation Dodell Wright s'engage pour la diffusion des savoirs scientifiques dans la société ici à Genève. Mon nom est Olivier Dessibourg, je suis responsable de la rubrique Sciences au journal Le Temps, qui est partenaire de cet événement. Vous avez peut-être vu les interviews qu'on a publiées dans nos pages ces derniers jours et qui vont encore paraître, en tout cas demain, pour l'interview de Svante Pääbo qui sera là jeudi soir. Donc, comme chaque année, vous le savez, nous avons le privilège ici d'accueillir à Genève des orateurs de renom, de prestige venant du monde entier. Ceci pour qu'ils nous présentent leurs recherches, pour qu'ils interagissent avec vous, qu'ils répondent aussi à vos questions. C'est un privilège assez important que nous avons ici de pouvoir les accueillir. Cette année, évidemment, on ne fera pas exception. On l'a déjà vu hier soir avec la présence de Linda Buck, prix Nobel de médecine 2004, pour ses découvertes sur les gènes des récepteurs olfactifs. Et bien sûr, ce soir, jeudi et vendredi, encore avec 3 conférenciers de renom. Tous ces orateurs s'exprimeront sur le thème de la conférence qui a été, le thème du colloque qui a été choisi cette année pour ces conférences, à savoir, vous l'avez ici, décoder le livre de la vie, la révolution génomique. Pourquoi ce thème? Pourquoi avoir choisi ce thème important? Pour en savoir un peu plus, je passe immédiatement la parole à Yvan Rodriguez, qui est membre du comité scientifique de la Fondation Wright et aussi professeur de génétique à l'Université de Genève.
Bonjour à tous. Je ne sais pas si ça fonctionne. Oui, là, ça fonctionne. Alors, dans le désordre, mesdames, messieurs, monsieur le président, monsieur le vice-recteur, chers collègues, peut-être monsieur le recteur, mais je ne l'ai pas vu. Je me réjouis de vous revoir ici pour cette deuxième conférence de ce colloque Wright 2016. Pour ceux d'entre vous qui n'étiez pas là, qui n'étiez pas présents hier soir, je rappelle en 2 mots que l'idée de ces colloques est de créer un pont entre la science, les scientifiques et le grand public. Non seulement de transmettre aux générations futures la flamme qui anime les scientifiques eux-mêmes, de naturellement favoriser la transmission de la connaissance scientifique, des concepts scientifiques de base, mais peut-être surtout de faire mieux comprendre ce que la science elle-même signifie, de faire mieux comprendre ce que la démarche scientifique implique. Cette démarche consiste en l'observation d'un phénomène, puis l'émission d'une hypothèse qui pourrait expliquer cette observation, puis d'un test, d'une expérience qui va évaluer la justesse de cette hypothèse. Un test qui sera effectué 1 fois, 2 fois, 10 fois, 100 fois, par des dizaines de scientifiques différents, à des dizaines d'endroits différents, à des dizaines de moments différents. Et en fonction de ces résultats, de conclure qu'on peut adopter l'explication du phénomène, ou alors qu'il faut modifier cette explication, ou alors que simplement il faut l'abandonner et émettre une nouvelle hypothèse. Et voilà peut-être un des plus importants messages à extraire de ces colloques, parce que c'est la compréhension, c'est l'intégration de cette méthode simple, mais qui bizarrement ne s'applique souvent pas en dehors des laboratoires, qui nous permettra à nous, humains, d'être mieux armés face aux nombreux charlatans qui peuplent nos villes et nos campagnes. Pour en revenir au colloque et à sa thématique qui s'intéresse cette année à la révolution génomique, c'est-à-dire à notre toute nouvelle capacité de lire le livre de la vie, les conférenciers ont été choisis, comme je vous le mentionnais hier soir, parce que leur domaine d'expertise est directement affecté par cette révolution. Nous avons écouté d'hier, ce lundi, le prix Nobel Linda Buck, qui nous a décrit comment l'identification de gènes pouvait nous permettre de mieux comprendre les mécanismes de base de la biologie. Elle nous a parlé de sa découverte de la plus grande famille de gènes présente dans le génome des mammifères et comment l'utilisation de cette famille à l'intérieur de nos cavités nasales nous permet d'apprécier en bon papé vaudois ou alors, au contraire, de ne pas ingérer un poisson qui aurait depuis longtemps quitté son milieu naturel. Dans 2 jours, jeudi prochain, Svante Pääbo nous parlera d'évolution. Il nous parlera de notre histoire, à nous les hominidés, et en particulier au séquençage du génome de nos très proches cousins, l'homme de Néandertal et l'homme de Denisova, et de ce que la lecture de ces livres de nos 2 cousins, de leur génome, nous apprend sur notre propre évolution, sur l'histoire de notre propre espèce. Et finalement, nous clôturerons ce colloque vendredi avec une conférence du philosophe et essayiste Peter Sloterdijk qui nous fera partager sa réflexion sur le futur de notre espèce, futur qui est naturellement directement affecté par notre capacité non seulement de lire les génomes, mais de les modifier. Aujourd'hui, nous écouterons le professeur Snyder, qui dirige le Centre de génomique et de médecine personnalisée de Stanford, et qui va nous parler de ce que la caractérisation génomique individuelle, chez l'humain, va modifier dans notre compréhension des pathologies et dans notre façon de traiter ces pathologies. Et voilà donc pour resituer en quelques mots la conférence de ce soir dans le cadre plus large de la thématique de cette semaine.
Merci Yvan. Comme vous le savez, donc, cette conférence est une conférence, mais elle vous donne aussi la possibilité de poser des questions aux conférenciers à la suite de la conférence. À ce stade, je vous demanderai de ne pas éteindre vos portables, mais de les mettre sur mode silencieux. Vous allez comprendre tout de suite pourquoi. Les mettre sur mode silencieux, évidemment, par respect pour le conférencier, mais ne pas les éteindre parce que cette année, outre les micros qui se baladeront dans la salle et qui vous donneront la possibilité de poser vos questions, on teste une nouvelle manière d'interagir avec le speaker de ce soir, à savoir Twitter. Si vous avez un portable ou un ordinateur ou une tablette, connectez-vous. Vous avez le nom du Wi-Fi de l'Université de Genève qui est Guest Unige. Connectez-vous, c'est libre d'accès. Et lancez Twitter et posez-nous vos questions à travers Twitter en mentionnant le hashtag, donc le dièse, ColloquWrite. Mentionnez bien ce code-là, vous posez votre question sur Twitter et Yvan, qui sera avec moi, les relayera, seul ou en grappe, au speaker de ce soir, Michael Snyder. Vous pouvez poser ces questions en français et en anglais. Elles vous seront répondues en anglais, mais vous avez tous reçu ou dû recevoir un casque à l'entrée pour entendre les réponses en français. Donc, encore une fois, en français ou en anglais pour les questions, que ce soit posées dans les micros ou par Twitter. Voilà, je m'arrête là pour ce qui est des housekeeping issues et je repasse la parole à Yvan qui va peut-être nous en dire plus sur l'orateur de ce soir.
Tout à fait. Alors nous avons aujourd'hui le plaisir et l'honneur d'accueillir Michael Snyder. Le professeur Snyder a obtenu son doctorat en sciences à l'Institut de technologie de Californie. Il a ensuite effectué un stage postdoctoral à l'Université de Stanford. C'est aujourd'hui un expert mondial du domaine de la génomique et un des participants majeurs dans très gros projets, un projet massif appelé ENCODE, en acronyme un peu étrange pour Encyclopédie des éléments d'ADN, qui a pour but d'identifier la totalité des éléments fonctionnels présents dans le génome humain. Michael Snyder a développé de très nombreuses techniques, des techniques qui sont aujourd'hui utilisées par beaucoup de laboratoires à travers le monde. Son travail est un travail de pionnier. Il a été, par exemple, le premier à effectuer une étude à large échelle de génomique fonctionnelle. Il s'intéresse particulièrement au profilage individuel chez l'humain, tant au niveau génomique qu'au niveau protéomique. L'idée est ici, entre autres, de pouvoir évaluer les risques de pathologie et de disposer de données précises pour suivre le développement de ces pathologies. Finalement, ce lien direct entre ces recherches et la santé, ce lien direct avec la médecine personnalisée, a mené Michael Snyder à créer ou co-créer de multiples compagnies de biotechnologie, Protometrics, Affomix, Excelix, Personalis et d'autres. Eh bien, elles ont été créées par Mike Snyder. Son travail s'inscrit donc tant dans la recherche fondamentale que dans l'application des résultats de cette recherche fondamentale. Je laisse maintenant donc la parole à Michael Snyder qui va nous faire voyager dans les génomes humains, mais aussi, j'imagine, dans les génomes de ceux dont nous sommes les hôtes, le génome des habitants qui vivent à l'intérieur de nos corps, et j'entends par là le microbiote, c'est-à-dire les bactéries, les levures et les champignons qui colonisent nos intestins, qui colonisent notre peau et qui nous permettent à tous de survivre. Dear Mike, welcome in Geneva. We are glad and honored to have you here. The floor is yours.
Great, thanks so much. Well, thank you very much, Yvonne. I do want to thank the Wright Foundation, the University of Geneva, and especially Yvonne for having me here. It's really a wonderful colloquium, and it's wonderful to be part of this. It's also wonderful to see so many friends I haven't seen literally in decades, so this has really been great. I'm going to talk about how genomics and big data we think will impact health and why I think health of the future will not at all be like health of today. I'm going to talk a little bit about science. I'm going to talk a little bit about personal journey. And I can't help but throw in some editorial comments that hopefully you'll find interesting. This is what frames our thinking in this area. We all know this, in fact, intuitively. That is your health. And the way we like to think of it, it's a state. Your health is really influenced by many things. Most of the time, we're healthy, but occasionally we do get into a disease state if something goes wrong, a virus infection, or more serious things like cancer, heart disease, what have you. And we know we toggle between these. And again, it's influenced by many different factors. It's influenced by your DNA, and then all these various things you're exposed to, which sometimes has the name exposome. So it's the food you eat, pathogens you're exposed to, life stresses, exercise. We know intuitively that all of these things will affect your health. And I would argue in the future, we want to understand this quantitatively, because in the not-so-distant future, people will be born with their genomes already sequenced, and we'd like to understand, with that genome sequence, what kinds of environmental exposures would lead to what kinds of health outcomes, what kinds of things will keep you healthy, and what to avoid disease. And this will be, I believe, in a probabilistic fashion of the future. So let me give you a little background on DNA. I know most of you probably know this already, but it's meant to bring all of us up to speed. But you probably know we all start out as a single cell, and we wind up with somewhere on the order of 10 to 30 trillion different cells. And we all don't come out looking like that. That's probably more than DNA anyway that's causing that. But the point is, it's our DNA that's a major factor in directing this to become that, to become a thinking, talking human being. So our DNA, I think many of you know, it's made of 4 chemicals or 4 building blocks, and they're strung together in a fashion, and we have 6 billion total of these building blocks all put together in an ordered fashion. And they're packaged into things called chromosomes. So we have 23 pairs of human chromosomes in every one of our cells. And so basically, our DNA is packaged into these. And for the most part, the DNA of one person looks fairly similar to that of another at the 6 billion base level. But when you actually start looking at it in detail, you realize that, in fact, there are, in fact, many differences. In fact, there are 4 million differences that are different between you and the person sitting next to you. That is single nucleotide, single chemical changes. So changes like this. And then there are larger changes as well. There's 500,000 of these, and there are even some big, big changes. Things of 1 million of these chemicals can be inserted or deleted that will actually differ between people. So we do have these changes, and they're called variants. So these changes are what makes you different, again, from the person sitting next to you. Unless that person's an identical twin, then you're very, very similar. You have very similar DNA. So it's now possible to find all these variants. The way this is done is there's some new technologies out there. I won't go into this in detail, but in the last 10 or 12 years, these new technologies have come along. So it used to be the very first genome costs about $1 billion to sequence. And now you can do it for $1,200. So less than $2,000 will sequence a human genome. Okay, that's actually what we pay right now for a research genome. That's just the sequencing. The interpretation costs $15,000 the way we do it. So the bottleneck isn't the sequencing anymore, it's the interpretation of the genomes, and that's something we're getting better. It will get much, much cheaper, but right now it is pretty expensive for a healthy person. Okay. There's other technologies I'll be mentioning later for the aficionados. There's been an explosion in some new techniques that use something called a mass spectrometer. This is what they have at airports that detect explosives. Well, we use fancy versions of this that let you follow now tens of thousands of molecules in your blood. And I'll be mentioning that a little bit later. So these two technologies basically let us follow human beings at a level that's never been possible before. This one's a little bit more mature, sequencing people's DNA. I forgot to say that your collection of DNA is called your genome. And so the name of the game is actually you can sequence an entire person's genome now with these new technologies. And again, it's $1,200. And because it's gotten cheap, you can actually now see that genome sequencing has become a very important part of medicine. That is to say, it's showing up now, and I'll talk about this in a little bit more detail. But genome sequencing is having a very important role in cancer, in solving undiagnosed, we call them mystery diseases. It's also true it's having an important role in prenatal diagnoses. So you can actually sequence a person's or a fetus's genome in a pregnant mom. So some of the DNA of a fetus actually circulates in a pregnant mom. And these days, there are official tests that you can do to find what are called aneuploidies, chromosome abnormalities, just by a blood draw from the pregnant mom. And it's technically possible to sequence the whole genome. That's not being done routinely for technical reasons, and also probably there's some ethical issues that have to get handled too. So it's being used in all these different areas, and one area that we're very useful is a somewhat related question: what if you're healthy? Is sequencing a genome useful to you? And so we're going to explore that tonight as well. I want to just give you a few examples of these first two cases. That is genome sequencing of cancer and undiagnosed disease. So starting with cancer, you may know that cancer is a genetic disease. So you can actually be predisposed for cancer, that is if you've inherited it from your parents, or you can get DNA changes that occur. Sometimes if you've been near nasty chemicals or too much radiation, that'll increase your chances. But we all actually increase chances just as we get older. So genetic changes occur as your cells divide, and actually as you go through life's journey, you will get certain changes. And so it's thought that in general, it's not typically one change that causes cancer. It's probably multiple changes, and it may be as many as 10 or 20 changes that will actually drive cells to grow out of control. And so the way genome sequencing is becoming important is that people are sequencing these cancers, and I'll show you how we do this in a minute. And the idea, it turns out that every cancer is different. That is to say, if you sequence one cancer, it will be different from another one. And so people are sequencing the DNA of cancer patients to try and find changes that might suggest a drug treatment. Okay? And so basically, typically you'll sequence the genome, and I'll show you this in a minute, and then you try and see what kinds of biochemical pathways might be suggested from this, and then come up with drug targets. I'll show you an example right now. So imagine you have cancer, and I've been through this with relatives, where basically you will actually sequence the DNA of the cancer cell, and then also typically of the normal tissue as well, blood or saliva you can actually sequence. and you actually sequence it quite deeply. And then you look for changes that are showing up in the cancer that aren't there in the normal person's DNA. And then you're trying to find these changes that might suggest a drug treatment for that person. So we did this initially. This is, in fact, the very first person we did. This is an individual that had colon cancer, and it actually spread. So metastasis means it spread out, and that's a bad situation. So cancer, when it's caught late, often is spread to other organs, and then it's very, very difficult to treat and deal with. What we had done with this particular individual was we had sequenced them. We sequenced their DNA from the cancer, and we looked for changes. In this case, we were looking for changes where they had extra copies of genes that might give a clue as to why they had cancer. And this individual, the way you'd look at this is that this is normal amounts of DNA, but they had 2 regions in their DNA that had extra copies. So extra copies means they're higher here. And this individual had extra copies of this one gene and extra copies of another region that had a different gene. Both of these genes you can actually target with a drug. You can target, actually it's the proteins made from them, but the point is that because we had sequenced this genome, we could actually see these genetic changes, and that suggests that we should use a drug that targets that one and not something else. This was a big deal because basically this particular type of cancer, it rarely has a mutation in this gene. Only 2% of the time it has that mutation, but this person did have it. And that was critical because we sequenced it, we saw it, and they did undergo surgery to remove the cancer, but then they got treated with this drug, and as far as I know, they've been cancer-free ever since. So the point is you can actually, again, match drugs to the particular genetic mutations to be able to better treat people. And because the mutations differ from one cancer type to another and from one individual to another, this is how the matches are made. And I can tell you personally, we've done this in my family because my mother-in-law got ovarian cancer. And so naturally, we went out to sequence her DNA. Her health was declining very quickly. And through some genetic tests, actually a mutation was found, and there was an experimental drug she went on, and it improved her health for about 9 months. And so for 9 months, she was doing fine, and then it came back. And that is a problem, which is even though people often respond to these treatments, usually in the long run, the cancer does come back. So you basically will, in the future, we're going to need multiple drugs to be able to treat cancer. But nonetheless, this is a whole new avenue that is now being used. And so again, you're going to see cases where drugs are used to match genetic alterations to be able to treat cancer. The other major area where genome sequencing is having huge impact is in undiagnosed or mystery diseases. So these are often called rare diseases, but in aggregate, they're actually not so rare. That is to say, it turns out in the US, and I think the numbers are similar worldwide, something on the order of 0.4% of children are born each year with a serious health defect. It also turns out by later in life, the number of people who show up with a genetic defect is much higher. A lot get exposed in teenage years, especially heart problems. Many of these are thought to be due to genetic changes, right? Because they're showing up early, they haven't had a chance to be environmental changes, not so much. And so what people are doing is half of these cases, you can actually make guesses what might be wrong with them, and people sequence their DNA, sequence a very specific gene, but half the time they don't know, and so people are turning to genome sequencing to try and solve these diseases. One of the first cases that was quite spectacular is this one here. This is an individual who was sequenced at Wisconsin Medical School. And basically, this is a child who was healthy until age 2, and then he got a cut that didn't seem to go away. And then more and more serious, actually, problems showed up, primarily in his gut, which later, in hindsight, turned out to be immune problems. This child had over 100 surgeries, had his entire colon removed, and actually his health was still declining very, very rapidly. So people turned to, this is the first case where they turned to sequencing this child's genome, and they discovered there was a mutation in this gene which actually causes an immune defect that made sense for the particular kinds of problems this child had. And so in the end, they could actually do a bone marrow transplant, give him good cells that repopulated his immune system, and he's been doing fine ever since. Here he is at age 6. I think he's 8 years old now. And so by sequencing this gene, he basically, in this case, led to a therapy that actually helped. Now, I can tell you, this only works 30% of the time. That is to say, 30% of the time, we can find out what's wrong with a child by sequencing their DNA. We do quite a bit of this. And I can tell you, the number of times it leads to a therapy is even lower. It's more like 2% of the time. It's very rare. But it's still very useful to find out what's wrong with the causative mutation, if you will, in the child, because it's a relief that parents have finally at least understand what's wrong with their child. So anyway, people are turning to sequencing. Now, in the other 70% of the time, this is a more common scenario. You'll sequence a child's DNA, and you'll get a list of genes that might be wrong, any one of which could be responsible for this. This is especially the case if it's things like developmental delay. They're slow in developing. And this child had intellectual disabilities and other issues as well, liver problems. And basically, when we were done sequencing the DNA, we found actually 8 changes, any one of which could have been responsible. And we were a bit baffled, but we were trying to slug our way through it to figure out which of these might be the cause of the mutation. The way this child got solved was actually a second child got sequenced over on the other side of the country. So we did this one at Stanford. This one was done at Duke University. And they had their child was somewhat similar to this one in characteristics. And basically, they thought this might be the cause of the mutation. And they put that information up on a blog, social media. And actually, someone was reading this and said, well, that's true. That's one of our 8 mutations. So in fact, because these are very, very rare, these changes, the chances of this being twice and not the same gene are almost not at all. And the bottom line is because of this match was made, it's a recurrent mutation found twice for a very rare disease. It was felt that this must be the cause of the mutation. And there are now 38 kids around the world, all actually found through social media and other means, that actually have a mutation of this gene and have fairly similar characteristics. So that's how this one got solved, of all things. And if you think about it, this is crazy, right? We're solving disease by social media. I mean, it does work, but it's not probably the best way to go. And this is the strongest argument for why we need to share this information, so you can put this out there. So imagine there will be a time when there will be millions of genome sequenced, and to be able to make these kinds of matches will be very, very valuable for solving human disease. And that's why sharing information in science is so, so important. So the lessons out of this part are, again, the success rate for figuring out the cause of a childhood disease, it's about 30% from sequencing their DNA. The information, as I mentioned before, it doesn't usually lead to therapies, but it usually helps solve the disease. For the parents, this can also be useful in planning their next child, because if they are going to have a second child, they may want to do in vitro fertilization. This does happen, and then select an embryo that basically lacks the mutation that caused the disease in their earlier child. So this is the sort of thing people are doing. And again, I mentioned that we need a very large database to do this. So you may say, well, this is great for solving disease. This is having impact, and it's true. What about me? I'm a healthy guy. Will getting my genome sequenced be useful? And this area was and still is somewhat controversial. A lot of people will tell you it's a bad idea to go around sequencing genomes of healthy people because you're going to turn everyone into hypochondriacs. I don't know what the word for that is in French. I hope that gets translated well. But the idea is you'll turn people into worriers, where they might wake up in the morning and worry from their genome about all these evil things. Well, we weren't so worried about it. We wanted to actually test this idea out. So the idea was, could you sequence someone's DNA, and then based on some other information, say medical tests or family history, you might be able to predict the diseases that they're at risk for, keep an eye on those diseases, possibly diagnose them early, maybe monitor diseases or treat them based on their genome sequence. This is the idea. Okay? We actually wanted to take this a step further, not just sequence their DNA, because there are new technologies I mentioned to literally be able to follow tens of thousands of molecules now in people's blood. So we actually did this. We did these incredibly deep profiles on people. Most of these words may look foreign to those of you who aren't scientists. But we not only sequenced their DNA, We actually sequence now all the modifications on their DNA. We follow the various molecules that are ultimately produced from their DNA. So we follow all their RNA, called their transcriptome. We try and follow all their proteins. We're able to follow about 2/3 of them using mass spectrometry. We have some very special tests to look for very important immune molecules called cytokines. We actually spend a lot of time on this. We've optimized a lot of procedures for being able to follow all the chemicals in your blood and urine, actually. So we follow metabolites and lipids. These are certain chemicals, very important chemicals. They actually carry out a lot of the energy functions and other signaling functions in your. So we follow those in blood and in urine. We also follow what your immune system is recognizing. And lastly, I'll talk about this a little more in a minute, but we follow the bacteria that live in and on you as well as other organisms. This is called your microbiome. And we follow this out of your stool, your poop, along with your nasal, skin, tongue, and urine. So we follow 5 different microbiomes, as it's called. I'll explain a little bit more about this in a minute. So we do these very, very deep analyses on people. Now you may say, why are we doing this? Well, the reason is we're trying to understand what it means to be healthy in incredible levels. Imagine we're literally making billions of measurements, and we're trying to see what does it mean when you're healthy, like while you're sitting there right now. And then what happens when you do get disease? If you get a disease, what does it look like? How does it onset, et cetera? And ultimately, how do you treat it? We want to understand how these different components relate to one another. We know that the bacteria in your gut do relate to all the metabolites that are in your blood and your urine, but we don't know the equation for that. And so as scientists, we want to try and figure this out. We want to understand how does your DNA relate to your immune system, relate to your microbiome, relate to your metabolomes, relate to all these different components and things. So we want to understand this in incredible detail. We want to understand how people react similarly and different from one another when faced with certain adverse conditions. So imagine I threw a virus on this room. Half of you would probably get infected, depends on the virus. And that half would probably all have certain common symptoms. That is to say, you'd probably get runny noses and certain things that would all be common, and certain molecules would all be the same. But you'd all probably be a little bit different, too. You'd have some personal responses that would differ from one person to the next. So we're trying to understand the commonalities as well as the differences. And, of course, I'd love to understand why one half didn't get sick. That's another issue, too. So by doing these deep profiles, we think we can do that, and you'll see in a minute. And then lastly, as we do these deep profiles, we think we can learn important information that will help the health of a person. And that turns out to be true. You'll see we're now following 100 people in incredible detail, and we've learned things that were very, very important for these people to know about, both from their DNA and from other things as well. So the way we started this, we actually started this on me, partly because it was a very complicated, people were very concerned about doing this kind of analysis on other folks. And I live in America, so you have to worry about getting sued. And so by starting on me, I didn't have to worry about suing myself. It was a very safe approach. So the bottom line is that what we do is we take my blood, my urine, my poop, all these things, every 2 to 3 months. And then when I get sick or something interesting happens, I actually take more samples. So for example, I've been doing this now for almost 7 years. And basically, I've given up over 200 of these samplings. And I've been through 10 viral infections. Lyme disease, I'll mention in a minute. I don't know how rampant that is in Switzerland. Probably not so much. Yeah, you have Lyme disease? Okay. Well, pay attention. Okay. And then the other thing, I've actually had a nasty bike accident last summer, too. So what we do is we take many more samples when I go through these adverse events. Okay, so through these viral infections, these are all common viruses. This is a rhinovirus, very common. It's the most common virus out there. There's respiratory syncytial virus. These are adenovirus, all very common colds. And I have another 2 years of data I haven't put up on the slide. But the bottom line is you can see when I get sick. So 9 months into the study, I had this RSV infection. We take blood as soon as I get congested. These are all days. The next day, 2 days later, 2 days, 3 days, so on. So we do dense profiling when sick. And then again, every 2 to 3 months when healthy. And then in interesting times like before and after China, things like that, that might be interesting. And then lately, I've been doing a lot of food tests. So I've been testing different supplements to see their effect on my metabolic control, and you'll see why in a minute. That's to be able to control some of my metabolites. So we've taken lots of samples over the last 2 years. So we started with my genome, and my genome made many predictions, a lot of which I knew about already from my family history and other things. So we've looked for changes that might suggest I'd get a very strong chance of disease. I'll talk about that a little more. This is a type of analysis where we look for complex disease, things like diabetes, schizophrenia, autism, heart attacks, things of various sorts. And what we do here is you can actually, I won't get into the details, but you can look at your genetic information. You actually have to sum up over all your genetic information. And for hundreds of diseases, I've only shown the top of the list, but for hundreds of diseases, you can predict whether you're more likely to get it or less likely to get it based on your genetics. Okay? And again, mine match pretty much what I knew about my history. So for example, as a European male, so I'm of European lineage, there's a 100% chance of getting one of these diseases. This is 0%. That's my starting point. And then blue line means I'm lower risk. Orange line means higher risk. So I'm at lower risk for obesity, and that made sense. No one in my family history has ever been overweight, going back to my great-grandparents. I'm at higher risk for coronary artery disease and heart attack. That made a lot of sense. My father had 9 brothers and sisters who lived into adulthood, all had either coronary artery disease or some sort of heart problem. Actually, all of them but one. And so that was increased risk. I have high cholesterol. That shows up here and here. So that all made sense. But there were some surprises. One is I'm at increased risk for basal cell carcinoma. I did not know about that. But it turns out I had my brothers and sisters out for what's called a family reunion, where they're all out visiting. So we took blood from everybody, of course, so that I could profile them. But I just happened to ask 2 brothers and 1 sister, what do you know about basal cell carcinomas? Because it's in my DNA. And they said, well, it turns out they each had those lesions, but they hadn't shared it. Why didn't they share it? Well, because you don't talk about basal cell carcinomas around the dinner table, right? At least we don't in my family. And so what happened, it was, therefore, in my family history, I just didn't know it. So what I like to say is that your genome is a better record of your family history than your family is, because you can transfer the information better if you know how to interpret it. We're still learning, but this is one way. The other thing that came up very early in this study, which was a surprise to me, was high risk for type 2 diabetes. I was not aware of that from any of my family members. And remember, we were taking all these samples and measuring many, many other things. Well, one of those things we were measuring, as you might imagine, was glucose. So it turns out my glucose was just perfectly normal, but ironically, because of my genome, I went to get a very fancy glucose metabolism test. And the day I went in for that was that day there. The woman looked at me. She thought I was strange. There's no way you can have type 2 diabetes. I didn't have a family history. She looked at me and said, you don't look like it. There's no way you can have. So I said, well, I sequenced my DNA. I think we need to look at this. And she reluctantly agreed. And my fasting glucose, surprisingly, came out high that day. And we remeasured. We thought, I was surprised too. Came out, same measurement. We measured it a week later as even higher, and this is a better measure for steady-state glucose. It's called hemoglobin A1C, and it's arbitrary, but the number that makes you diabetic is 6.5, and I was 6.4. I was right on the edge. Okay? So 5 weeks later, I didn't know anything about type 2 diabetes because I was not aware of it from my family history, so I went to see my general doctor, general practitioner, measured again, came out higher, 6.7, classified as diabetic. That was April 11, 2011. Why do I remember that date? Until then, I had the world's worst eating habits. I ate plenty of sweets. I ate sodas, lemonade, things like that, because I didn't know I had an issue. And I cut it off from that day. Once this test came back, I said, I'll cut that out, which I did. I had been biking, but I doubled my biking, and I started running, and I gradually brought it down to normal. That's about 8 months. That's not a few days. That's a lot of effort. Okay, so this made a big deal at the time because it's the first time someone used their genome to predict their risk for a disease, and in my case got the disease, and was also manageable. So I could get it under control by changing my diet and lifestyle. So it was also very interesting because when it occurred right after what was a very nasty virus infection, this respiratory syncytial virus. And so the hypothesis is that I'm genetically predisposed for type 2 diabetes, and then in conjunction with this viral infection, that's probably what triggered the disease. We don't know that for sure, but that's the idea. Now, there's some critics out there who say, Mike, that might have gone away on its own. You don't know. So we accidentally did the right control. Okay? As we scaled to look at 100 people, we were collecting the data, but nobody was looking at it. So it was blind study. This is what you like to do in science. And this is my glucose again. This is my hemoglobin A1C. It had been marching along perfectly fine. Well, we stopped looking at it somewhere in here, and then right here somebody looked at it and said, Mike, your hemoglobin A1C is 7.0 again. You're diabetic. And so I had no idea, so he sampled me a few times. I started running, and I got it down to 5.7, but these days it hovers between 6.1 and 6.4, except when I got Lyme disease, it went up again. So the bottom line is there's no question I have a metabolic disorder, and it's, it's in my, it's there. I'd stopped running, so it might be due to the fact I stopped running. It might be due to this virus infection. I switched statins. Any of those could be responsible. It's not clear why it went up again, but it's up, and I can't get it back under control. So I'll probably go on metformin, actually, as a way of getting it in better shape. So, but again, there's no question I have a metabolic control problem, and it's not just me. My brothers and sisters were all elevated for these hemoglobin A1C, and they're just as skinny as I am or even thinner. So that is to say, we have this problem running in our family. And again, my genome is the thing that revealed this. Now, I think I've gotten behind, but I will say by making all these billions of measurements, we could look at my molecular pathways that were changing at a level that's never been done before. It's like getting an IMAX movie of seeing what's going on with your biochemistry. We can see in detail what was going wrong when I was getting type 2 diabetes. And we could see various biosynthetic pathways were actually all being misregulated that were very relevant to my condition. So we're learning about how disease forms using this kind of study. Now, we talked about this last night. Genetics is important, but another area that's just as important is epigenetics. And in fact, many of you may know that the things you do for your lifestyle, the food you eat, exercise, they all impact your DNA. And it's not fully understood how this works. It's something we're trying to understand. But basically, your DNA can get a chemical modification. It can get changes on it. And again, it's done by lifestyles, and scientists don't really understand how this works. We're trying to work on this. But we do know, I'll tell you one of my favorite things, that the nutrients you eat will actually affect several generations, especially during pregnancy. And then lifestyle factors. One of my favorite studies is an exercise study where a group in Sweden had 23 people bike with one leg but not the other for 4 months, 4 days a week. Okay, then they looked at the DNA in the biking leg relative to the non-biking leg, and sure enough, there were 4,000 changes in their DNA using a very primitive technology that could measure this. Okay, so what does that mean? That means when you bike or exercise, you actually are directly affecting your DNA. We know that actually affects gene expression because changes in your DNA caused by this chemical modification affect your gene expression. And so this is technical, but we've gone in and actually for about 20 of my times have actually mapped out all my modified DNA, and we can see some very interesting changes as well. I'll just tell you one interesting story, which is by looking at my modified DNA, not just my DNA now, where it's modified, I can actually see whether the genes are turned off by other means. So for example, I have another number of regions in the genome. This gene, which is responsible for making some important immune cells called eosinophils. It turns out this gene, one of my copies, is inactivated by genetics, and another is inactivated by this methylation, this chemical modification, which comes from my mom's copy. And so the bottom line is that this gene is actually inactivated by 2 means, one genetically, one epigenetically. And I don't have many RNAs from the good copy. Most come from the bad copy, meaning the good copy is not expressed well. And so once you know it, I have higher numbers of eosinophils, it turns out, as well. So I would argue in the future, I forgot to ask, how many of you want to get your genome sequenced? Okay. Many more in front than in the back. I don't know what that means. Okay. I would argue you not only want to get your genome sequenced, you want to get your epigenome done as well, too, because this is going to be very, very important. I think I've gotten a little bit late, so maybe I'll do this pretty quickly. But the other area that's very, very important, we're okay? Okay. The other area that's very important is your microbiome. You may have heard about this. But your microbiome are the bacteria and other organisms that live in and on you. And there are 10 times as many of them as there are your own cells. Okay, so we're mostly bacteria. Okay, so I'm talking to more bacteria than people in this room, by 10 times as many. Okay, it sounds gross, but it's not. These bacteria, most of them are in your gut, but they're in your skin and other places as well. They're essential for digesting your food. They make essential vitamins. B12 is an incredibly complicated molecule. They make folate, other things as well. They're all done by the bacteria in your gut. These days, the microbiome is implicated in every disease you can imagine, although what's cause and effect isn't so clear. These cases I've listed here, it's actually the evidence is getting quite strong. So inflammatory bowel disease, which is fairly frequent. Actually, a lot of this is probably due to your microbiome. Diabetes, obesity, very interesting experiments there showing obesity being due to microbiome. Even heart attacks has been implicated in microbiome, and autism, some very interesting evidence there. So we've also now been studying this as well on me and other people, as you'll see in a minute. Just for fun, we picked one very interesting time. So I mentioned all these viral infections. Well, I had one that was particularly intriguing. This was a rhinovirus infection. At least it started out that way. And then not shown here, but 9 days later, I got a second fever. So when we started to analyze the microbiome, we said, well, let's see if we can figure this fever out. So we sequenced my microbiome, my nose, my gut, my other areas. And we actually looked at all the bacteria and viruses that were there. By the way, we looked for viruses during that time, and I couldn't find any. So when you sequence your nose and your gut, you actually get hundreds of organisms that you'll see, and you just see which ones are there and how do they change. And this might be a little bit complicated. For hundreds of organisms, I'm only showing you the top list, we can see which ones are there. This is a time when I'm healthy. This is a time when I actually was sick with this mysterious infection. This is 2 days later, and back to healthy. And the amount of bacteria that's there is indicated by the color. So yellow means there's a lot. And the bottom line is that for this, one bacteria correlated perfectly for when I had this mysterious infection. This is from my nose. And it was Streptococcus. So the odds are for the Streptococcus infection, that this time I had this mystery infection, it was a Streptococcus infection. And this is probably the world's most expensive way to have figured this out, by the way. Okay. But it's a nice proof of principle, and it makes a point I'd like to make, which is that it turns out, right, when you get sick, nobody ever figures out what it is. Oh, you had the flu, 2 days later, it's better. If it doesn't get better, then they figure out what it is. It's a week later. They say, oh, it's mono. And then by then, you've infected 50 people. So if you think about what we should be doing every time somebody gets sick, we should be figuring out what it is. So if it is something serious, you can quarantine them before they infect 50 people. So I would argue the assays are getting cheap enough. This one was expensive, but you could do this for $50. And we would know instantly what everybody's getting sick from when they get sick. Again, if it's serious, you could treat it appropriately instead of waiting for it to get worse and spreading. The other area that's huge these days, I don't know if this is as big in Europe, but are these wearable devices. How many of you are wearing a wearable device? Fitbit? Oh, this time they're all on the back. I don't know what that means. Okay, well, anyway, these are getting very popular in the US. I use 8 of these devices every day. I have 2 smartwatches right now. I think in the future, your smartphone will be your nexus of health information, because most of these devices, they relay information directly into your smartphone. It's not all integrated yet. That's something we're working on. But these are powerful because they're collecting continuous measurements. Right? When you go to a doctor, they measure things in about 15 seconds. But these things are measuring you all the time. In fact, the devices I have collect 2 million data points every day. So we get a lot of information, and we can see what's going on. So why is this useful, you may say? Well, it turns out this is really valuable for when I got Lyme disease last summer, 2015. It turns out I was, well, let me start the story by saying I was in rural Massachusetts where they have lots of Lyme disease. 2 weeks later, I was flying to Norway, where they have almost none. And it turns out, you may not know this, but when you get on airplanes, your oxygen drops. This is very relevant for the Wright Foundation because they made the oxygen sensors, right? So it turns out every time you're on an airplane, you may not realize this, but your oxygen drops because they don't pressurize the cabin all the way to regular temperature. It's about 7,500 feet, so 2,500 meters. So anyway, the point is it drops, and when you land, it comes back. Well, I was flying to Norway, and it dropped, but it dropped much lower than usual, and it didn't come all the way back when I landed. And not only that, my heart rate, which goes up on flights initially too, it actually remained elevated when I landed in Oslo as well. So I knew something wasn't quite right because I measure myself all the time. I knew it wasn't jet lag. So the bottom line is then I got a low-grade fever for 4 days. I went to a doctor in Norway, and I said, I warned him ahead of time it might be Lyme disease because it didn't seem normal. And he measured my blood, and sure enough, my immune cells were up. And so he said, yep, that could be bacterial, it could be Lyme. I think you should take penicillin. I said, I think I should take doxycycline. It was a little tense for a few minutes there, as you might imagine. But he gave in and he took doxycycline, and sure enough, it cleared it right up. You take it for 2 weeks. And when I got back, I actually tested, and sure enough, I was positive for Lyme. I have the perfect control for the scientists here because it was negative before I left because I'd given blood. And I could just test that sample. So it's very, very clear I'd gotten Lyme disease, and these wearable devices were very powerful for figuring this out. This won't mean much, but we've now written algorithms, it's a great name, called Change of Heart. It's based on heart rate, so we can tell now whenever you get some sort of inflammation illness, like a viral infection or what have you. And it's because you can pick up outlying heart rates and now skin temperature. Turns out, this we found after the fact, but your skin temperature, not surprisingly, goes up as well, or at least the number of outlying measurements for skin. So we're now doing to 100 people exactly what we're doing to me. We're looking at them in incredible detail. So we sample them every 3 months when they're healthy. And then when they get a viral infection or other stress response, 6 people had colonoscopies. We sample them more densely, either before, after, or during these times. And we're also running a lot of people through dietary perturbations, but I won't have time to talk about that. So we're trying to see the exact effect of food on your biochemistry. So again, we do dense sampling during these periods. What's special about our study is that all of our folks have agreed to make their data open, which we think is very valuable for the reasons I mentioned before, all but a few. So the data are going to go out in public, so actually the scientists here can analyze this. And again, these are going to be incredible data sets for the scientific community to look at. This is where we were at the beginning of summer. We're about 1,500 sampling so far. Well, you can guess who number one is. That's me, obviously. But we have many samples from lots of other folks as well. So what can you learn from a study like this? I can't tell you everything, but I'll give you a few vignettes. Let's start with the genome sequencing. It turns out, if you start looking at people's genomes, so we've sequenced them all, we've analyzed 48 of them in incredible detail, just like mine. This study, by the way, has been going on for 3 years. So we've looked at the genome, and 8 of them, we found something that's important that they have to watch out for. And sometimes this is really important. One example, 2 people have a mutation in this gene, which puts them at high risk for this particular kind of cancer. One of them, I know for sure, did not have a family history, so they were not aware of this. And so basically now there's a simple urine test they'll do every time they go to the doctor to look out for this. Another individual, I won't have time to go through all these, actually turns out, I forgot to say that 65 of them are either pre-diabetic or elevated or at high risk for type 2 diabetes. I have a new interest in type 2 diabetes, as you might imagine. So anyway, this one individual, it turns out he was thought to be elevated for type 2 diabetes. Turns out he has a mutation in a different kind of diabetes. It's very different from type 2. His treatment is 100% different from what you would do for type 2. It's called MODY. So it's a very different kind of treatment. And this was, again, discovered from his DNA. We'd not have known this had we not sequenced his genome. This is another interesting case. This is an individual we sequenced. We just finished our sequence. And this is a lesson for the future. And then he came up right after we sequenced. He said, Mike, my father has a heart attack, had just had a heart attack in his 60s, and he discovered his uncle had died of a heart attack. He knew he died, but he died of a heart attack in his 50s, and another individual, a close relative, had died of a heart attack as well. So basically, he said, can you look at my genome? And we found this mutation, which means that, first of all, we think we found the cause of the mutation, and second, he's got it. So he did actually it's called a stress echo. It's a stress test you do, and sure enough, he has some abnormal heart issues. And in fact, now he's on drugs because of this. And so we found this, again, by sequencing his genome. So I think your genome will not only be interpreted at the time you get it, but will also be important in the future. So if you learn new information, you'll go back to it like a reference, like a book, and you'll see if you can see any information there that might help your health. And that's true in this case. So this is very important information for him to have learned. All right, I think I'll wrap in a few minutes, but we actually have a number of individuals who are doing, I mentioned, different kinds of dietary studies. We're doing exercise, we're doing supplements. We have a bunch of people that are actually doing weight gain, weight loss, what happens when you gain and lose weight. So remember, a lot of our folks are pre-diabetic. They're actually have something called insulin resistance, which is thought to be a precursor to type 2 diabetes. Most folks who are insulin resistant will become diabetic in their lifetime. So 13 of these individuals are going through weight gain, weight loss, and 11 weight-matched healthy folks have gone through this as well. And we look at the changes that are occurring when they do this. It turns out, even at baseline, before they gain and lose weight, we can tell the insulin-sensitive and the insulin-resistant folks apart, the ones at risk for type 2 diabetes from those who are not. Okay, so we're trying to understand what makes pre-diabetics different from normal people so that we can try and actually identify it early and treat it properly. And it turns out these insulin resistance, these pre-diabetics are elevated for these genes in gene expression by RNA. We can show this from metabolites as well, other very interesting. And the normal folks are more elevated for these. So the bottom line is we can tell apart, these are biomarkers they're called, differences between those who are pre-diabetic from those who aren't. And the goal is to try and catch disease early. We need to validate this in a larger group. We've also looked at people just at normal baseline. So we look at healthy people. We look just how similar and different are they. And it turns out everyone is different. So everyone is special and not just to their mother. Okay. So here's what's shown here. These cytokines are very, very important immune molecules. At baseline, meaning before they have any disease or anything, we all have different levels of different immune markers. So these are 63 cytokines. Each color is a different person. And so this means there's a lot here, and in there means there's a little. But the blue person has a lot of IL-7. The light brown has a lot of this one. The orange one, this one. So everybody has different levels of these immune markers, which we think actually predisposes people differently to different diseases. Okay, that's the next thing to test. But the point out of all this is we are all different, even at baseline. And we think these kinds of measurements will be very, very powerful for seeing this apart. And lastly, remember the microbiome I told you? This is looking at people's stool microbiome. It turns out that if you go through a weight gain, weight loss, or if you get a viral infection, your microbiome will shift. So your stool bacteria will shift. But you actually still look more like you than anyone else. So for a short-term change, your stool bacteria will shift, but they still are personal microbiomes. And I think that's because we tend to eat the same food. If you actually have a very dramatic long-term shift in diet, you can shift your microbiome. That's been shown. So the point is that we have personal microbiomes. We think it's very important for digesting our food. Where are we going with all this? Well, this is the idea here. We want to see how different people, when faced with different perturbations, say, some people eat bad food, they get overweight, they lose metabolic control. But plenty of people actually stay perfectly healthy eating the exact same foods, and we've seen this already in our food stuff. That is to say, in fact, there are some people who are incredibly obese, but they're not diabetic at all. They actually have good metabolic control. They may have other issues, but at least metabolic control is fine. So we'd like to understand that difference. We also want to understand how different people, when they get viral infections, some get sick, some stay healthy. I got type 2 diabetes after my RSV infection. We think it's because I'm genetically predisposed. Most people don't get diabetes after that. This is Mike Snyder's world. I don't necessarily think this is everyone's world. But I'm a believer we will be getting genome sequenced before people are born. We're going to have other kinds of biochemical information. It's not as deep as what we're doing to me and these 100 other people, but we'll still have deep profiles. We won't be measuring 15 things in the doctor's office in the future. We're going to have wearable information, and with that, we're going to be able to better predict risk, diagnose disease, et cetera. So the bottom line is personal genome sequencing is here. all of you will have the option of getting your genome sequenced. So it's not, whether it's not, can I get my genome sequenced? It's whether you want to get your genome sequenced and what you might learn. Kind of curious, how many want to get their genome sequenced now? All right, more hands. I had a positive effect. All right. I think the genome's just the beginning. I think we want these other biochemical analyses as well. I think you want to do this in a longitudinal fashion, meaning you want to get yourself analyzed now while you're healthy because you want to see your deviation from baseline. You want to see your deviation from a healthy state. And to me, that's why it's so important. You don't want to compare when you get sick, your sick state to everybody else's healthy. That's hard to interpret. You want to see your sick state relative to your healthy one. I mentioned everybody's profile is different, but I also am a strong believer in the future. No one's going to understand you like you will. So physicians of the future actually aren't, currently they're not set up for this. Physicians of the future will be coordinators of healthcare, not dictators of healthcare. And I think that's how that'll roll out. And so I've had an amazing team of people, I won't have time to go through this, have really helped out. I will mention Tracy McLaughlin's been my clinical collaborator, George Weinstock on the microbiome, and just a really talented bunch of folks in my lab. And then here is a very shameless plug for a book that we had that just came out. If you like this kind of area I've been talking about, this is actually written for the educated layperson and physicians, so it's to get people comfortable with this space. So feel free to look at it. It doesn't cost very much either. It's not meant to make me money. It's just really to get this information out. So I think we can now take questions if people have any. So thanks again for having me.
Thank you. Yeah, thank you, Michael, for this fascinating talk. Take a seat. So we now have 3/4 of an hour to talk with Michael. Maybe my first question, you want to take some water?
Yeah.
You told us you're measuring yourself regularly, right?
Constantly.
How much time, how much energy, how much attention does it require from you every day, and what do you do with the data?
Okay, it does take a fair amount of time, mostly in charging the devices. Every morning I have to get up, or every evening I'm plugging these things in. So that's probably the biggest part. Right now the data are mostly automatically collected, and they go into a common database that we have. And then it takes us a long time to interpret it. Because this is also new, we don't have it working on the fly, so to speak. That is to say, we have to collect the data, download it, and then have teams of specialists analyze it. But in the future, I think it will become automatic. So much like, we only picked up the Lyme disease because I was following that so closely for those parameters, but the biochemical measurements often takes us several weeks or months to get that information.
And attention, I saw you yesterday evening at dinner. You were looking at your iPhone like every 5 minutes.
Well, I don't think I look at it that often. I was probably showing people all my wonderful gizmos, so.
Okay,
but I do look at it periodically. Airplanes is what I look at. It's very fascinating.
Yeah. Let's come to this idea of diagnostic. You said it, right? You don't want us to become hypochondriac. That's the word in French, too. But still, until now, diagnosis was a one-shot event, right? We go to the doctor, we get a diagnosis, diagnosis, and then we go away. Now with this new technologies, it's not a one-shot event. It's a continual, right? It's continuous. So how does it change our relation to being sick or being healthy?
Yeah. So I think first of all, it's going to change things dramatically. I believe medicine of the future will be different and should be different. So let's back up a little bit. Right now, what do? People go to the doctor once they're sick, once there's a problem, right? Occasionally you'll go as a healthy person. I don't know about you, but in the US, probably go about every 2 years if you're a healthy person. So first of all, that means you don't discover things until after the symptoms arise. And what we're trying to do is find things before that. We're trying to predict what you're at risk for and catch things early, which makes all the difference. Cancer's a good case. If you catch cancer after it's spread everywhere, it's very, very difficult to manage. But if you catch it early, breast cancer is a good case. You catch it early, you can actually use surgery, and then people will be fine. Or these days, people do prophylactic. You may know for breast and ovarian cancer, they'll remove the breasts and ovaries as a means of preventing cancer. So it will shift to a more preventative thing, and that's going to be good. So, but to address other issues, it is true that if you go around sequencing people's genomes, you have to coach them properly. We have genetic counselors. It isn't, well, I would argue everyone should get their genome sequenced, but everyone shouldn't necessarily get their information back if they're a worrier. So if you're a worrier, you may want to not get all your information back because you don't want to wake up every morning saying, oh, am I going to get breast cancer or going to get autism or something like this. I think what you really want is for those who are comfortable with it, you return that information. And also for those who maybe they want some information like that, I would argue BRCA, maybe people should learn about regardless. Again, it's still your choice, but you may want to learn that because you may want to take action on it. And there's a few diseases like that that probably should go back right away. But the other thing I'll flip around, I'll add one last point. People are very comfortable using family history in medicine. If you go to a doctor, they always, first thing they ask is, so what's running in your family? Diabetes. And people are very comfortable with that, yet they're scared of genetics, and that makes no sense to me. Because if anything, genetics is probably better than family history for trying to use the information for predicting risk, and so I think we should just be adopting it. That's my own feeling. But again, it's up to the individual to decide.
We have those powerful tools, right? Genetic tools to get early diagnosis. You mentioned the so-called liquid biopsy, the idea being to search for markers in the blood's mother's blood to in the fetus to like Down syndrome, for example. It's getting common.
Yes.
You didn't mention CRISPR-Cas9. This is a really powerful technology to edit and change the genome, right? So the question is, will we still have the right to be sick? Will we have to feel guilty when we're sick because we have the tools to diagnose this and the tools to change our genome to cure it?
Wow, that's a big one. Let's see. Should we feel guilty about being sick? Well, I think we've got a long ways to go before we worry about that. I think we get sick from viral infections and things we have no control over. Although it'll be very interesting. I think in the future, we will have sensors on us that detect viruses from other people, and that will get very interesting. Yeah, as far as correcting things, same thing. I think we're a ways from doing gene editing for correction unless you already have disease in certain cases. So that is being used in cases like AIDS for trying to affect the DNA of the blood so the virus doesn't spread very well. So this is one potential therapy. So that is coming out. Germline editing is still, it's in the too early days, I would say. We don't know how safe it is and things like that. And the ethical issues have to be debated. Although I think in the long run, there won't be any issues about trying to fix disease-causing mutations. That's not a problem. I don't think anyone will have an ethical issue with that. Where it becomes tricky is when you start trying to affect what I call enhancement traits, like trying to make smarter people or more athletic people by changing the DNA in embryos. I think that will get very controversial and obviously needs to be debated appropriately. But I think for curing, evil diseases, I don't think that's a problem, at least not for me.
We'll now open the floor to questions, so please ask for the mics, and please use the Twitter account. Just recall it, #ColloquWrite to ask your question. So maybe first question, can someone bring a mic down here? Yes?
En contrôlant si bien la génétique, on doit pouvoir maîtriser la maladie morale qui fait tant de ravages sur Terre, y compris provoquer des cancers.
I heard the first person. I can control genetics, but I should also control...
Like controlling genetics, one should also be able to control moral illnesses. Or
Control illnesses. Sorry, mine's actually a little...
I can help you with translation if needed.
Oh, I should control morals? Well, moral illnesses. Oh, boy. I don't think they pay me enough for that. I don't know what to say. I think, the ethical issues are certainly very real, and they certainly need to be properly debated. Again, for me, curing disease, that's my primary goal. I have no issues with that. you know, what are we going to do about people whose behavior seems a little strange? We wouldn't want to eliminate all of that. You know, it's a fine distinction between people who have Asperger's and go on and produce incredible things for science, and those who might be a little bit tipped further over and have difficulty functioning in science or functioning in life. So that is a delicate trait. So I don't know that I would have want to go in and mess around with that. These are important, you know, ethical issues to discuss.
Another question here, Yvonne. Can you give the mic just behind you?
Thank you. One general comment to thank you for this very stimulating presentation. I think it can inspire generally for the possibilities not only and mainly to reduce pain for people and make them happier and healthier, but could also help improve health services, reduce budget and redirect it to more relevant thing and reduce contamination. This is the general thing and very inspiring. Second question, you mentioned autism. Speaking about need for repetition of sequencing, how frequently and if there's any event only of health or otherwise. And second, is there a possibility or already envisaged of using this sequencing not only to see the risks, but also help in the administration of antipsychotic medicines, which are very destructive and very little use or help, and the use with other tranquilizers. The effects are dramatic and very, very little is done because nobody knows what and how. And it seems to me that what you mentioned, after a period of crisis, blood is taken anyway for checking many things, for anemia or other disturbances. So that would be really the crux of a problem, which could improve the use of medication, follow up, and changes.
Sure.
For a very dramatic problem.
Yeah.
Thank you.
So with regard to the first question, I mean, I think people generally get their DNA sequenced once. It does change in your different cells as it divides, but those are fairly infrequent. So I don't see us getting our genome sequenced very many times, although I see us getting our epigenome sequenced a lot. And in fact, I didn't say, but you can actually tell someone's age from their epigenome, from their methylation. It works perfectly. Like, mine's spot on. I wanted to be 25 years younger, but it's spot on for my modified DNA. With regards to the drug responses, I'm not an expert in the neuropsychiatric drugs, so it's hard for me to comment on that. But I do follow a lot of some of the other space, and there are getting to be lots of groups that are sharing information. So one problem with the US is the medical information is very fragmented. It's a mess. And so the US is way, way behind the rest of the world, where they have single-payer systems and can actually share information much more readily. Like, so in Canada, they can share their medical records, and there are now big projects out there to map DNA. They're not quite as far along on the sequencing yet, but they will be able to map drug responses to people's genetic information. There's quite a bit of that already, and there's something like 200 different genes that have been involved in drug response. And I guarantee if you don't learn anything else when you get your genome, you will learn some useful information about your drug response. So my genome tells me which statins to avoid that I might have an adverse side effect on, for example. And it tells me if I ever have a heart attack, what my dose of warfarin and some other blood control drugs should be, at least for starting medications. So your genome does have a lot of useful information. It's actually one of the best studied areas is the drug responses, except I'm not as sure about the neuropsychiatric drugs that you're bringing up. So I think we should collect all that information. There will be a world, again, where there'll be millions of kids with their genome sequence and who are taking drugs, and we should just collect all that information and see who responds to what with these diseases. I'll add one last point, but I'm happy to take another question while you get the microphone over there maybe. But it turns out all these complex diseases, they're probably not one disease anyway. They're probably, like diabetes is probably many, many different diseases all lumped together because they all have high glucose. And so we treat them all the same, yet we know they don't work. If you take type 2 diabetics and you give them metformin, 80% will respond, 20% no way. Likewise, 20% will respond to salicylate, the rest don't. So it's clearly operating differently, and I think part of it's because these are different diseases, and autism's a great example of this as well.
We'll come back to you, but maybe are there questions on the web?
On Twitter. So there are quite a bit of questions. One that comes back a few times is, when do you think that the omics analysis that you presented will be available to a larger public that is not restricted to the lab?
Yeah. So this is a research project. It costs us $170,000 per person per year. So we're not ready for it now, but that's because we're doing incredibly deep profiling. I think a lighter version of this can be done for a few $100s, so not as deep. And my own prediction is some form of it's coming out now in a fairly light fashion. There's 2 companies in the US and some others that are forming that I know of that want to do these, at least have several hundreds, maybe a few thousand data analyses associated with people and sample them more frequently than what we currently do. So some form of this is starting to roll out already. I think on the wearable stuff, that's going to catch on like lightning. That'll just go really, really fast. Because as soon as you show it has utility, people want it. And in that case, it's simply a matter of writing the algorithms to get it all integrated into a system that basically, you don't want to sit down and figure out 8 different devices every day. You want it all to go into one thing, and a red light goes off when something evil is happening, like Lyme disease, maybe an orange light when it's a rhinovirus infection, and then maybe yellow if you're feeling a little congested or something, but green means okay. Now, in the US, you have to worry that these things are all matching certain standards, so they have to make sure they're medically validated and approved by the FDA, or you see a whole series of regulatory issues. So that may take time. But still, I think it'll happen in a few years for the wearable devices.
Monsieur?
Merci beaucoup. Merci beaucoup pour cette passionnante conférence. J'ai une question toute simple. Les assurances médicales, qu'est-ce qu'elles vont faire avec toutes ces datas qu'on leur fournira dès notre naissance, voire même avant notre naissance, et ensuite en permanence par le réseau Wi-Fi ou des choses comme ça? Est-ce qu'on ne risque pas de voir ce progrès nous revenir dans la figure? Parce qu'elles mettront des limitations, parce que vous n'avez pas le bon gène, donc dommage, on ne prend pas en charge cette maladie-là. C'est de votre faute. Ou alors il faut que vous fassiez que vous mangiez que ça et on va surveiller ce que vous mangez, sinon on vous sort du système. Voilà.
So this is a really good question. I don't know how it works in Switzerland, but in the US, you cannot be discriminated on in health insurance or in employment based on your genetic information. So in principle, you're not allowed to. Again, I don't know how it works in Switzerland. You are allowed to be discriminated for life insurance and long-term disability in the US. So that still does exist. Now, I would argue it's better to know you're at risk for these things than and catch them rather than let them go and have problems, and I'll pay extra insurance if that's what it takes. Now, long-term disability, I disagree with. But I would actually just turn this entire thing on its head. That is to say, I think insurance companies should pay you to get your genome sequenced. Why? Because you're going to use that information to better manage your health. And that's true for me, because I can catch this stuff early because I have my eye out for it, because I know what I'm at risk for. And I think that should be true of every human in the genome, every human on the planet. I think they should be paying us to have our genome sequenced. I would also flip this a little bit, which is I would argue, it's the job of every civilized country to take care of its citizens at some level. And you guys are in better shape than we are in the US. We still have lots of people who have no health insurance and who are in trouble. But we're obviously trying to correct that. We'll see what happens tomorrow where we stand with all this. But the point out of all this is that we should have some minimum level of health care for people in any developed society, I would argue. And so I would like to see that happen. That's sort of separate from your genome sequence.
Michael, allow me a follow-up question on that.
Sure.
How do you see the arrival of internet giants like Google? I mean, they're very active in that domain. How do you see this happening regarding privacy, security of data, and so on?
Yeah, well, you probably know Google is very interested in the health space now, as are these other countries, Microsoft, even Amazon, because health is basically an information science. So it's a matter of learning things, learning what's in your DNA and these other measurements, and trying to actually figure, again, how to better manage people's health and also drive prices down. So you could actually set up financial models as to, at some point, how do you value life? That's a tough one in terms of financial terms. Although people have done this. I don't know what I think of that. But the point out of all this is that you can actually model this stuff out, though, and figure out what these different treatments cost and what kind of health benefit you can do. And you can actually set up models so that, in fact, hopefully everyone wins at the end of the day. That is, the insurance companies that cover you, actually, if they know what your costs are going to be, they're going to need a profit margin built in, no question. But you can model out what things you will pay for and which ones you won't. By the way, for very expensive drugs now, you can actually get reimbursements from your insurance companies for cancer drugs, because if they're so expensive, they'll do the sequencing first to see if you'll respond for certain drugs, rather than the opposite. So in that case, that's one of the few examples where they know it saves money.
Are there questions? I see a question there, but are there questions in the back? There is one in the middle. I'll come back to you. Can we bring
Bonsoir. J'aurais voulu savoir sur l'épigénétisme, de un, s'il était réversible, et s'il ne l'est pas, s'il est transmissible à une génération suivante.
So the answer is, can it be reversed and put on next year? I don't know if it can be reversed, but I sure hope so. I want to figure it out for me before it's too late, because aging is very epigenetically affected. Can it be passed on to the next generation? Absolutely. There's some very, very fascinating studies. For example, here, I'll give you one, where it turns out in Gambia, there are children that are conceived either in the rainy season or the dry season. And it's when they're conceived that counts. And if it's in the rainy season, there's less food, and those kids come out shorter, and it passes on. So at least that's the mom, when they're pregnant, is actually having an effect on the baby. And there's plenty of cases you may know from the St. Petersburg siege that had long-term effects. So during World War II, there was a siege of St. Petersburg, and there was a lot of starvation, a lot of people died, and they actually lasted several generations. You can trace that in the grandkids. So these effects do go on several generations. Now, we don't know all the details of how that happens, but it'd be fascinating to know.
I have a question on the aging and anti-aging movement. And you mentioned the supplements, that you made an experience with nutritive supplements. What do you think of measuring deficits and optimizing your health by taking supplements or hormone regulation and hormone supplements your whole life, so that you add optimization to the genetic what you explained? Thank you.
Yeah, I don't know enough about hormone supplements, and I would worry about some of them because you've got to get the doses just right, and also where you are in certain times. And I mean, I know a little bit more about the postmenopausal stuff. But I think as far as supplements, it's a good area to explore. It's one we are exploring. And the reason I'm doing that, it's actually hard to change people's diet, right? Lots of people get overweight and obese, and you can tell them you've got to lose weight or you're going to get diabetes and a heart attack, and they don't. But the one thing everybody does is they do take supplements pretty easily. So that's why we're actually trying a lot of supplements for 100 people to see what effects they'll have. And we're seeing some interesting results, but it's too early to talk about right now. So I do think it's a promising area because I think it's something people would actually adhere to.
We'll turn back to Twitter for a while.
So there is an interesting question on Twitter, which is, which are the pathologies that will be the less affected by knowing our genomes?
Oh, which pathologies are less affected? Boy, there's a lot of tough areas right now. You know, the more complex neurological diseases are still difficult. We are learning about, so things that are early in life are more likely to be due to a genetic cause. And so those are the ones we have our best chance for. Things that are later in life have been more trouble. Like we know aging is due to genetics. That's a complex situation. But we don't know what all the genes are that are responsible for this. It's very complicated. So I think the later in life diseases are very hard, and the more complicated they are. So I think some forms of diabetes are going to be pretty tough to figure out as well. So those are some that come to mind, I guess. Yeah. There's one in the way in the back.
Other questions in the room? There's one up there and then one here.
My name is Naginder Sani. I'm not a scientist, even less a genetician. But having listened to your superb presentation, I feel like a genetician. And so I'm worried about myself a little bit, but not much. I feel quite healthy. I'm probably healthier than ever before in my life. I'm soon 80 years old. I never ran more than a kilometer until I retired at the age of 60. I started running, I don't know why, and now I can run half a marathon. Often I wonder if you know that a lot of long-distance runners come from Kenya, from a place called, or near around a place called Eldoret. That's where I was born. Now, do you think I inherited a gene because I was born in the same area where the long-distance runners come from, and that gene woke up when I was 60? Could sequencing me would help to get my history, and that might be useful in your study. Thank you.
Yeah. So you've covered a lot of ground there. First of all, there are genes associated with athleticism, so some that affect muscle mass, things like that. I don't know anything about, well, no, that's not true. There are some genetic loci that have been suggested to deal with increased pulmonary artery capacity, so that is lung capacity. I don't know enough about Kenyan long-distance running genetics, although I think it'd be very fascinating to look at this to see if there are any special genes in that group. The fact that your gene woke up at 60, I imagine it was there the whole time, but I just think you probably chose to focus on that when you did so at your time in your life. That would be my own guess. I think it would be incredibly fascinating to sequence. I think anybody who lives a really old age is super interesting to sequence because we want to understand why you're not dead. That is to say, imagine you hit 100 and we sequence you. We want to know what it is that makes you so special. And there are people out there who should have serious disease. They call them resilient people, who have genetic changes that say they should have a certain disease, and they're doing just fine. So they're very special people because they must be doing something that's suppressing that effect. And we'd like to understand that. So as you keep going along in your very healthy state, we really would love to look at you someday. And there are groups out there that are doing this. There's called the Wellness Project. They're sequencing people who are 80 or over just to do that, just to understand why these folks are so healthy and they've never had disease.
I'd like to ask you a question about health, and one important person when we are talking about health or sickness is our doctors. Actually, now the doctors are not trained, right, to this revolution. So is it realistic to ask them to be trained on that, because we know how tough it is to become a doctor, right, already? Or will we have like new jobs in the future, which could be like genetic counseling, so special doctors that would go to just to ask questions about our genomes? do you think about this?
Yeah, absolutely.
Should general practitioners be trained to answer questions? I had my genome sequenced. I went to my doctor. He was totally unable to answer my questions.
Yeah, no, I think you're hitting on a really good point. So physicians are not prepared for any of this. And people are showing up in the United States with things like 23andMe. It's a genetic testing test, and they have no idea what to do with this information. So we do have to train doctors in the future to at least understand what is going on here. It's actually one of the motivations for writing the book I did. So they do have to be aware of it. I don't know about Switzerland, but in the US, you spend 15 minutes with your doctor when you see them. So there's no way they have time to understand you the way you can understand you. And I really think this is why you have to be in charge of your own health. So I think you'll probably collect a lot of this information in your smartphone. It will show up. It'll be automatic process. The doctor will probably look at some things and say, he'll be more the coordinator of healthcare, I think, in the future instead of the dictator of healthcare. I really think right now we train physicians to act like they know everything, when rarely do they, especially in 15 minutes. You just can't know enough to give information back. And so what happens is they have to act confident and say, well, usually they tell you exactly what you know already. They just give a fancy name. So I had some nasty rash. They said, well, you have contact dermatitis. Well, I knew that already. They just called it contact dermatitis. It was a rash. So we need to be able to go beyond that, where we can do these tests and really understand depth. I'm sure I'm offending lots of people here, so you can clobber me later, I guess. But I really do think what they'll do is they'll see this information, say, well, based on this, you should see this person or that person, and a genetic counselor will be one of these people who will actually be the ones interpreting your genome. So I think we're going to bring a lot more information, and it's going to be good for everybody, it's including the physician, because the physician's going to show up with a lot more information at their disposal to make a much more accurate diagnosis, and I think that's where we're going to go.
So time's running, but we have time for two, three more questions. There was one there, yes? You have a mic?
Bonjour, je suis désolée, je vais parler en français parce que je ne sais pas parler anglais. Merci beaucoup pour votre conférence. À un moment, vous avez parlé des prédispositions qu'on pouvait voir dans le génome et vous avez parlé de schizophrénie. Et du coup, je voulais savoir comment est-ce qu'on peut voir une maladie mentale dans le génome.
Yeah, the way you see it is that there are certain genetic changes that are associated with this. For complex diseases like schizophrenia, it usually takes several changes to have a chance of seeing an increased risk. And it's not a perfect situation. That is to say, you'll see these changes and you'll say, well, you'll have an increased chance. Much like my type 2 diabetes, I went from a 26% chance to a 47% chance of getting type 2 diabetes. So it's not perfectly deterministic, but because it was such a big increase, it had me on the alert for it. And I think that's what's going to happen for schizophrenia. Schizophrenia is a lot more complicated. It turns out autism, I don't think, is so far away. I think we will be making fairly accurate genetic predictions, at least on some forms of autism, in the not-so-distant future, because there's less genes involved, which makes the predictions a little bit easier. I don't know if that was clear enough. But anyway, it's by looking through the DNA and seeing these changes that are associated with individuals who have either schizophrenia or autism.
Twitter questions?
Yes, is there anything like too much information?
Yeah, in my world, the answer is no. It might be for some people. I mean, it is true, we literally have 0.5 petabyte of data on me. I don't know if you know what that means, but let's see. Do you guys know what a gigabyte is? So, all right, so you go from a gigabyte to a terabyte to a more. Well, no, to terabyte to metabyte, and then I guess exactobytes next after that. Anyway, my group of people, we have several petabytes of data. It's a lot of information. But at the end, you process it and you distill it down, much like we don't know how all the parts of our car work. Maybe some of you do. But we know how to get in and drive it. We get the relevant information back to us. And I think that's how it's going to come back. We'll get this information. It'll be processed, so we'll say, well, this biochemical pathway is up, this one's down, and therefore you might have this or something else. And I can tell you this really does work. I mentioned the Lyme disease example, but there was one individual in our study who we actually discovered had early lymphoma by actually doing all these detailed measurements on them. And again, that was very useful. That's an early form of cancer, blood cancer. And so that was very useful to have found out, as you might imagine, for that person. So I think that's how it'll show up. You'll see these early signs, and these different sorts of information will be processed in a fashion that the orange light will go off or the red light will go off, and that will tell someone, let's do the following follow-up tests. And that's why I think it's going to be powerful for the physicians, because it's going to give information that they'll use to do the next test, to see things in more detail.
We're going to take one last question from the audience. Yes?
Yeah, thank you very much, and thank you again for the great insights you shared. Going back to the slide you had on the gut and understanding about how many more diseases are related to the gut, well, how do you see the consumption of some supplements and probiotics in the future being affected by this?
I think it's going to be hugely affected. So, well, first of all, you probably know you guys look much fitter than in the US, but we eat 500 times more sugar than we did even 60 years ago. So diet has changed dramatically for the worse. And so we've shifted, and with that has, we think, changes in microbiome have occurred as well. But one thing we're learning now is that all the stuff is individualized. That is to say, if you eat a certain kind of bread, we can see what it does to your glucose levels and also to your microbiome. And that will be different from the person sitting next to you. So we're going to see individualized diets that will affect your microbiome, which will affect your metabolism, which is very important for you. And one reason for this, it turns out your microbiome is very intricately related to your immune system. So your gut and your immune system are in this balance all the time. And your immune system is absolutely critical for your health as well. So all these things are in this balance, and food is the engine that's driving this. So, well, food in your genome and your epigenome. So you can't do much about your genome. You've got it. But you can do something about your food that will shift your microbiome, it'll shift your immune system, and it will shift your metabolites, which will directly affect your health at all those different levels. And again, I think supplements are a big part of this. And so it's a matter of understanding which general supplements we should be doing, but also which supplements are exactly important for you and not the person sitting next to you.
There's one very last question here.
Last but not least, for those of us who now do want to get our genome sequenced, is there a bad or a good place to go, like a good and a bad doctor? How do you actually choose and go do it?
Okay, well, that's one that'll get me in trouble. You can get your genome sequenced for $1,000. I have no money at stake here with Veritas. They're a Boston-based company. They will give you back your sequence. How good that interpretation is, I'm not sure. I'm a bit skeptical. I know they don't go through genomes the way we go through genomes, which takes 40 hours of person looking over this. So that's why it's so expensive. That's why it's $15,000 to interpret a genome. You can also go to Illumina. They will also give you your data back. Again, I think they give you what I would call a light interpretation, and that's how I get myself in trouble. I'm going to get all kinds of tweets off this thing. But the point is that you can get some information back. To get a very deep one, it's going to take time, and you'd probably have to get in very special studies. There aren't that many groups that go as deep as we do for interpreting genomes.
So I'd like to close this discussion with talking about the future, right? One day we might go and settle on Mars, the planet Mars. So why am I talking about Mars? We're talking about genomics. You might have heard of this story of those two twins, Mark and Scott Kelly, American astronauts. One went for 1 year on the International Space Station. The other one stayed on Earth. And you are going to analyze, I think, who was it, Mark or Scott? I don't remember.
Scott was in space.
Okay,
Mark
Scott,
was on the ground.
Scott's DNA to see what damages has been done by the cosmic rays and so on. Can you tell us a bit more about this? If you have already some results, what have you found in
yeah, well, we've only collected all the samples and we're processing them now, so we don't have any real results. But we have collected, just like we were doing for me and these other 100 people, we were actually collecting samples on Scott, who was in space, Scott Kelly, and on Mark Kelly, who was on the ground. And we're actually following their biochemistry as Scott, as he went after blasting off in space. And the whole year he was in space, we're sampling him. And we'll see what kind of biochemical changes he has.
What do you expect?
I think we'll see some very interesting stress responses. I predict we will see differences in his epigenome. I don't know how much difference we'll see in his genome. He is getting blasted by radiation, but it's a little tricky to see those changes. There'll be some, but we'll see how much.
I'm sure we'll hear about it in the next months or years.
Stay tuned.
Thank you very much, Michael Snyder, for a fascinating conference. Thank you. Voilà, cette discussion va certainement se poursuivre ces 2 prochains soirs. Donc, vous avez le programme sous les yeux. Jeudi soir, Svante Pääbo sur l'homme de Néandertal et vendredi, Peter Sloterdijk. Il me reste à remercier la Fondation Wright pour l'organisation de ce colloque, à l'Université de Genève pour l'accueillir, à vous tous d'être venus, à tous ceux qui l'ont mis sur pied aussi pour leur travail. Et je vous donne rendez-vous jeudi. Il reste une dernière chose. Si vous avez aimé cette conférence, si vous l'avez détestée, si vous l'avez trouvée inintéressante ou très intéressante, dites-le nous. On organise un petit sondage. La Fondation organise un petit sondage pour améliorer les prochains colloques. Donc, prenez 5 minutes, ça dure 5 minutes, pour nous dire ce que vous pensez de ces conférences. Merci beaucoup. À jeudi. Au revoir. Thank you very much.
Okay, very good. Okay.